Shall we start? Huh? Yeah. So, last time we talked about, so we first started talking about Hardy-Weinberg, where we said that nothing changes basically. Which means that there's no microevolutionary processes operating. That is mutation, migration, selection, and drift, right? As well as there's random mating, which is basically no dissociative mating, etc., etc., right? No variance in reproductive success, etc., etc. Okay, so these are the conditions under which Hardy-Weinberg is satisfied, and Hardy-Weinberg, as Kavita pointed out, equilibrium is achieved in one generation. We then spent a little bit of time talking about migration and drift. So migration, we talked about most simple model, which is basically a continent island. Yeah, and we talked about unidirectional gene flow, and we just wrote out a simple algebraic equation which says how the allele frequency here will change over time, right? Then we talked a little bit about drift, where we said drift is because of finite sampling, because we're violating this assumption of infinite population size, right? So we talked about how drift would operate and what the impacts of drift would be in terms of loss of heterozygosity, as well as variance in allele frequencies across populations. So there's two ways to think about drift, right? One is that if you think of two alleles in a population, P and Q, and then you had to pick out some n individuals from this population, then based on this binomial formula, you can talk about how many times you might expect to get, 5 P's and 10 Q's and so on and so forth, right? Each of those outcomes has a probability. So in that same vein, if you think about, thousands of populations of the same size, even they might have different distributions or numbers of alleles, right? The P and Q may be, a number of P and number of Q would be different. That distribution is just governed by this, that is drift. At the same time, if you look across time, drift would result in the loss of heterozygosity because you have a possibility of inbreeding or sampling the same individual again. We talked a little bit about inbreeding as well and how inbreeding will make heterozygosity decrease and increase homozygosity, right? We also talked, so that's basic. And then we talked a little bit about FST, how you will measure this. So maybe, I don't know whether this is all getting a little muddled up, but maybe we'll try to sort it out. Like the way at least I understand it is, in these forces can be quantified algebraically or stochastically, how they are changing, how change is happening over time, right? Those are basically deterministic in most cases, unless you're talking about drift. Change is only going to happen in a specific generation and exactly, right? You can exactly predict what the change in allele frequency will be. Except for drift, which is stochastic, you can predict an expectation though. It's not like you have no clue what will happen. You can have an expectation of what the distribution of allele frequencies will be under drift, right? So it's still predictable, right? And that's fine. And these are all still, we've still not talked about selection and mutation, which we will today, But really, evolution or population genetics happens with everything together. So if you look at empirical data, if you look at real populations, really how genetic variation is distributed between populations, that need not be, I mean, it's hard to bring that to this framework. So often we use statistics to quantify things, and we link the statistics to the parameters. In this case, for example, FST is a function of N and M, right? N and M are used in our equation about how allele frequencies will change, right? So this is how you kind of link the two. If it seems like they're very different worlds, data or empirical observation is a very different world from these models or theory. These are not models, it's just kind of algebraic changes in allele frequencies. So now selection. So selection is very easy in some sense. And basically, remember we had 3 types of alleles. Say we had A1A1, A1A2, the heterozygote, and A2A2. And remember, this was P square, 2PQ, and Q square. So why are we writing these genotypes, by the way? We're talking about genotypes in the case of selection because selection acts on the phenotype. Okay. So hopefully there's some link between genotype and phenotype. We'll talk about that in a bit. But assuming there is, the only way to quantify selection is through the genotype, right? In some sense. I mean, you can quantify it based on the phenotype, but then you have to know the link between the two. So basically, suppose you had differential selection on these 3 genotypes. That is, imagine that, remember that first day we did that bag with the beads. So imagine the pink beads were a little sticky, right? And you basically, when you put your hand in, you had a higher chance of your hand will just stick to the pink bead and you'll pull it out. So you can think about this W11, W12, W22 as also, in some sense, biased survival, right? Some alleles which are, individuals which are A1, are more likely to survive than those which are A1, A2. In our other Hardy-Weinberg, we just assumed that W11 equals W12 is equal to W22 is equal to 1. That's what we assumed. But when you're thinking about selection, the model then basically assumes that this is not the case and there's differential fitness of these 3 genotypes. Okay, W11 may not equal to W12, may not equal to W22. Okay, so now you know that basically these are the relative fitnesses. You can easily calculate after selection what proportion of A11 individuals will survive. Huh? Yeah, P square W11, right? 2PQ W12 and Q square W22. Fine. But it could be the population size is changing. Say W11 is more than 1 or something like that, right? So you have to normalize for the fact that the population size might change. So you calculate something called W bar, which is basically P square W11 plus 2PQ W12 plus Q square W22. Okay. So now, if we are thinking about, say, delta Q, after selection, how does Q change? Right? That's what we're interested in. After selection, how does Q change? So this basically Q1 after selection minus Q0 before selection. So what is Q1? What is Q after selection? What is Q? Q is the frequency of A2. How do you calculate the frequency of A2? Louder. Q square W22 plus half 2PQ W12. Correct? Yeah? Okay? Divided by W bar. Because you have to normalize for the fact that the number of individuals itself might be changing. Okay? So now then delta Q is equal to Q square W22 plus PQ W12 by W bar minus Q. Right? Okay? So this basically you can multiply by W bar, so it becomes Q. If you simplify this, basically you can do this, right? And if you simplify this, delta Q is PQ into Q W22 minus W12 P into W11 minus W12. Okay. So that's, if you simplify that, that's what you get. So what is this saying now? That's fine. That's some algebraic equation. But what does it mean? What can we understand about selection and what happens with selection? So let's forget the formula, okay? Let's forget the formula for now and let's think very simply. Suppose I told you that W11 equals W22 equals W12 equals 0. Sorry. Let's make it simpler. W11 equals 0. W12 equals W22 equals 1. What kind of a model is this? I mean, what's happening, like biologically, what is this? Huh? Okay, homozygous is lethal. Yes, homozygous is lethal. What can you say about the genetic system here? Huh? A2 is dominant. Okay, good. So this is a situation of dominance. It's not an additive trait where the phenotype is intermediate, right? So it's, okay, so what will happen in this situation? What will happen to allele frequency in this situation over time? Which one will get fixed? A2 will get fixed, right? So you don't need to know this formula, right? If you just think about it, like heuristically, you can have an expectation of what will happen. So then let's think about it in this way. Okay, so what happens in each of these quadrants? Okay, so what's happening here? W22 is between 1 and 2. W22 is more than W11. Okay, so what will happen over here? Okay, let's look at this one. This is the easiest, right? This we just kind of, well, we just kind of explored this. Here, W22 is more than W11, right? Depending on whether it's 0 and 2 is the most extreme example, whereas 1 and 2 or 1 and 1.5 or something like that would be slightly less intense differential selection, right? We're talking about differential selection here because there's a difference in selective pressure on A1 and A2, correct? because we're only talking about W22 and W11. So what will happen here? You already said it. A2 will increase. Correct? Right? Right? Is that clear? No one's confused about that, right? What will happen here? The opposite. A1 will increase. So what will happen here potentially? Both W11 and W22 have fitness less than 1. You can make an assumption about W12. W12 is the fitness of the heterozygote. So what if W12 is greater than W11, which equals W22, which is less than 0? Less than 1. Huh? Yeah, but what is that called? Heterozygote advantage, right? The heterozygote has advantage here. And so what frequency will that go? By the way, this one will eventually result in fixation of A2. This will result in fixation of A1. What will happen here under heterozygote advantage? Huh? Why? Yes, half-half, but why? Heterozygosity is highest when the frequencies are equal, right? So if it bounces off of that, it will come back, right? So in this case, you have heterozygote advantage, and so you will end up with equal P equal to Q. What about here? Here what's happening? W11, W22 are more than 1. Okay. And W12, I'm telling you, is less than 1. Huh? Loss of heterozygosity, but then if you lose heterozygosity, those others are advantageous. Yeah, but what will happen to the final? Can you predict finally what the allele frequency will be? And it will be the same exact thing every time?
What is your initial allele frequency? And
yeah.
That will actually go to either A1 or...
Yeah, so you could have A1 fixing or A2 fixing, right? So it will kind of depend on where you start off, right? Deepa, hi. So this is heterozygote disadvantage, okay? But the outcome will be fixation of either, right? So can you think of an example of this? Real life, I mean, nature. Yeah, sickle cell anemia, everybody says with their eyes closed, right? Okay, that's heterozygote advantage. What is the heterozygote there? What is the advantage?
So they are more susceptible to malaria, right? So in that manner, they have less.
Yeah, please use.
So I guess it's the case in Africa where there is too much malaria infection also. So if someone is having good blood type, so malaria parasite basically so means malarial parasite basically grow on good type of blood cells. So that will get round. So, wild type blood cell type. Okay, so malarial parasite grow on that, so they can they are more prone to get malarial infection and they can die. And if someone is having sickle cell, so malarial parasite cannot grow on it, but they have a disadvantage that they have sickle cell anemia, which is also not advantageous to your health. But if you have heterozygote. So you have advantage that you are compensating for a bit in your health, but malarial parasite cannot grow on your blood cell.
It's a nice story. Do you know about, there's malaria not only in Africa, there's a lot of malaria in India, right? and Southeast Asia. So is it the same here? Did you know that sickle cell anemia is actually very common in India? Do you know where it's common? Think about it. There are populations which have very high anemia, sickle cell anemia. Anemia in general is very common in India, right? So, huh?
But that when you have anemia, but that is a different due to dietary reasons.
Yeah, but I'm saying that sickle cell anemia is also quite common in India. There are other diseases in India which are also common, which are associated with malaria. Similarly, not necessarily only sickle cell anemia. Okay, so you can read about that. But what about this? Think of a hypothetical situation where you may have advantage to the basically to this allele. How much time do I have? Oh, these things. Think of a situation, no? Lethal, someone said. You said. Give an example of a lethal. Anyone? These are genes, right? They have to be examples. Huh? Hemophilia is not lethal. And it's also sex-linked, so there's some complications there. We have not talked about sex-linked genes at all. Any housekeeping gene? Hey, come on, no, come on. No, this is a very random answer. So give examples of lethals.
Again, arbitrary example, but maybe some recessive lethal disease.
That's it. Defining the question. Yeah. Okay, you should look it up. There are lots of, yeah. Some developmental genes. He said housekeeping genes, but yeah. We don't always know about lethal mutations because the individuals are not there in the population. But possibly, if you have an enzyme, a mutation in the active site or something like that, which makes it non-functional, those kinds of mutations would be lethal, right? Okay, so I just wanted to show you one thing, which is, yeah, someone said directional selection also, na? What's directional selection? Yeah, someone said stabilizing selection. Why is this called stabilizing selection? Yeah. Okay. Why is this called direction? Where is directional selection here? Okay. Heterozygote advantage is also called overdominance. I'm sure you know that. Yeah, so just to basically, so the basic selection model which we discussed assumes following about the genetic system, single biallelic autosomal locus, right? Diploidy, random mating amongst individuals. We also have assumed that selection is identical in both the sexes. Selection occurs only through differences in viability. What happens if selection is occurring via differences in reproductive success? Viability means survival, na? We are talking about differential survival. But it could also occur through differences in reproductive success. What's that called? Sexual selection, yeah, right? That's sexual selection. Selection is also constant in space and time for each genotype. It's not changing. W11 is W11 is W11, whether you look today, tomorrow, or the day after. So the environment is not changing in this. There's no fluctuation changes in the selection pressure. We also assume non-overlapping generations, no mutation, infinite population size, no gene flow, right? And no inbreeding. So all those are the assumptions we're making in this selection model. So basically, just to give you an idea, so if you have A1, A1, A2, 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 you have W11, W2, W22. So you can have recessive lethal, right? Which is 1, 1, 0, right? That's an example of recessive lethal. You can have detrimental alleles, where you have a recessive situation, where you have 1, 1, 1 minus S. Why is it? It's detrimental because the fitness is a bit less, right? It's not lethal. So it's 1 minus S. S is a number less than 0, right? Oh, sorry, S is an integer. So it is 1, S is a number less than 0. So it could be 0.8 or 0.6 or something like that, right? And then you could have an additive system. What would happen in the additive system if this is 1 minus S? What would be the fitness here? What minus S by 2? Got it correct? Understood? Because it's additive, it's midway, right? Okay. Then you could have dominance, which is basically purifying selection. And here you can have, you can have dominance, you can have both purifying and positive. Purifying. So purifying selection is 1 minus HS, 1 minus S. So this is a special case of this in the sense that it may not be S by 2, but it could be, S by 3 or S by 4 or something like that, right? This definitely has a higher fitness and this is less. Or you can have positive selection where you have 1 plus S, 1 plus HS, and 1. What's the difference between these two? In this case, A11 has a higher fitness, higher than 1, right? That's why you have the W bar. You just make everything normal because, with that you can normalize the whole thing. These things don't have to add up to 1, right? That's why you have W bar. And then the last ones are, I'll write it here, heterozygote advantage. What's the general form of heterozygote advantage? What would be the general form of heterozygote advantage? 1 plus S, good, right? Does that make sense? And heterozygote disadvantage, oh, by the way, this you could make it, you could also write it as 1 minus S1, 1 minus S2, heterozygote disadvantage, 1 plus S1, 1 plus S2. Understand? So that's just a very general framework, and then basically you could plug in to that formula and always calculate change in allele frequency. Yeah. H, it's just some, yeah. So this is how, for example, suppose I sent you that paper, but hooks, yeah, sorry. Yeah. No, heterozygous advantage. It doesn't matter. It's just that these two, if you write it like this, you can very easily compare the two. You can write it any other way. I mean, it could be 1, 0.5 and whatever, doesn't matter. Just that relatively, those two are higher than the one in the middle. But if you write it like this, you can compare advantage and disadvantage because you have S1 and S2 in both. So, for example, in the real world, do you know selection coefficient S? You might do an experiment where you look at differential survival of, say, white mice on a black surface versus a white surface or something like that, right? That's how you would estimate selection coefficient. Well, that's not necessarily selection coefficient, that is just differences in viability, right? But what you could do is, suppose you are tracking a population over time and you saw some change in allele frequency, right? So you could say delta P over X generations equals to this. And then you could you would have to guess at what kind of selection is happening. And then based on the model, you can try to estimate what S or H or any of these things are. So it's not trivial to study selection in the wild. And obviously, when you have selection on traits which are on sex chromosomes or something like that, it will become much more complicated, sex-linked, yeah. This one? Dominance. Yeah, it's just about, basically, in the net fitness here is less than 1. I mean, but basically this might be, you might be losing individuals, whereas here it's advantageous. Here it's disadvantageous, okay? Because this is less than 1, whereas here it's positive selection, so it's advantageous, but they're just flips of the same, you're right. It's just that in this case, it's easier to talk about it as one is purifying, which is decreasing frequency, and this is advantageous, so it's increasing. So basically, purifying selection decreases the frequency of detrimental alleles, whereas positive selection increases the frequency of advantageous alleles. So here the frequency will decrease, and here it will increase. So you're setting up the models in such a way, but there isn't much difference. These differences are mainly from the perspective of the biology. Like in one case, you'll increase the frequency, in other case, you'll decrease it. Something will always increase and the other thing will decrease. That's true, but. Okay, so basically selection is quite intuitive, a little bit easier than something like drift. But tomorrow we'll use that simulation package to just basically explore these things. And I'll send you the link today. We can do it on my computer. But you'll see how each of these different forces act independently and what happens when they act together, right? So what's really important is to get an intuitive understanding of how, well, it depends on what you're doing, but if you are working with empirical data, it's really important to intuitively understand how these forces might differentially impact allele frequencies. So now we spend a bit of time on mutation. Mutation is really simple, okay? So mutation is you have some allele P which is converted to some allele, say P prime. The rate of this is U and the rate of back mutation is V. Okay? That is, P is becoming P prime at a rate U and P prime is going back to P at a rate V. Okay? So then, what is the change in P? How many P's are there? Huh? What is it? How are you getting P? Generating P? Sorry? This is delta P. So how are you losing P? Yeah. So basically, U into P, right, minus V into Q. Sorry, this is going to Q. V into Q is adding to P. Yeah, sorry, this is delta Q, right? So basically, U into P is becoming Q. What's the matter? Oh, I'll have to go there. Huh? So yeah, so this is Q and this is P. So U proportion is of P is becoming Q, right? And V proportion of Q is becoming P. So if you look at how is Q changing, you're losing Q. How are you? You're adding to Q. U times Q is adding to Q, right? But you're losing Q because of V. So does this make sense? Why is this? U minus, why am I writing that? How did I get that? Sorry, one second. Why is that? Because it is 1 minus, P is 1 minus Q, right? So if you, basically it is U into 1 minus Q minus VQ. So U minus UQ minus VQ, U minus Q into U plus V, okay? So basically, in terms of the equilibrium, right? So for what is the equilibrium frequency of the mutation? The mutation is Q, right? So basically, then delta Q equals 0. So U minus Q into U plus V equals 0. U equals Q into U plus V. Q E equals U by V plus U. Okay? So generally though, these numbers are really, really small. Mutation rates are really, really low. How do you even estimate mutation rate? I mean, I just made up something. I said, okay, this P is converting to Q at some rate. How do you estimate mutation rate? We talked about the selection differential, that S, right? And we said we can estimate S by looking at differences in viability or looking at change in allele frequency over time. So how would we estimate mutation rate? Yeah, so that's one way, or you can have many what are called mutation accumulation lines, where you can have many replicates, like say in bacteria or something like that. And then you can sequence the whole thing and look at mutations and accumulation of mutations. So there's various ways to do it. But also very interesting and very important is that mutations and their fitness effects are not uniformly distributed. So what do you think most mutations are? Are they good? Do they have a positive S? Do they have a negative S? How is fitness distributed as a function of frequency? What kind of fitness effects do new mutations usually have? Huh? No effect. Deleterious.
Maybe it depends on where we are considering it.
Yeah, but this is a theoretical thing. No, in this you're not talking about, you're saying some mutation rate, some mutation. In the model, you're not modeling different parts of the DNA and all that stuff, right? So you need a general estimate as well. So actually, a lot of mutations are slightly deleterious. So that's the most common. So this is 0. This is W bar. Okay, that's the relative average fitness. Sorry. So these are kind of lethals, right? Because their fitness is 0. These are fitness is more than W bar. Advantageous. And how about all these? Yeah, they're slightly deleterious. And these are, yeah. Yeah. Neutral and slightly deleterious. Neutral and nearly neutral, sorry. Like up to here, say, whatever. And there are several which are slightly deleterious. Sorry, I can't hear you at all.
I don't understand this part, but you say that it's slightly different between the peak and the...
Yeah, so basically if this is, it depends on where the 0 is, no? So these are kind of more than 1. Oh no, sorry. This is relative fitness. So these are all nearly neutral. These are advantageous, right? And this whole spectrum is slightly deleterious to nearly neutral to neutral, okay? So let's do one problem which was where we kind of calculate viabilities. So Kuru is a prion disease, an unusual disease where the infectious agent is an abnormal protein found mainly in a tribe in New Guinea highlands. The women and children of both sexes had a tradition of eating their deceased relatives at mortuary feasts. Kuru killed approximately 1% of the population annually, but a ban on cannibalism in the 1950s resulted in a decline in Kuru incidence. Okay? The polymorphism at codon position 129 in the prion protein, PRNP, influences the susceptibility of human prion diseases. Mead et al. Examined women aged 50 and older who had a history of multiple mortuary feasts. The frequencies of the sequence that had M methionine or V valine at codon position 129 was 15 expected under Hardy-Weinberg. Where is this? Was nearly equal, 0.484. However, there are only 7 homozygotes, significantly below. So here, wait, 1 second. Genotype frequency of the 2 groups. Okay, let me just write it down. Okay, so here. So there's unexposed to feast. Okay? Exposed to feast. So why is that? Why am I writing exposed to feast and unexposed to feast? That's before selection, right? This is before selection. And this is potentially after selection. What selection? What's the selection? It's a disease, right? This Kuru disease. And some people are predisposed to it and others are not. The ones who are predisposed to it have a specific mutation. Okay? So you want to now calculate what's the selection on that mutation through viability, right? So here there's 140 and 30, and then you have genotypes. MM, MV and VV. What is M? The genotypes of the variant position 129. This is methionine and valine. Fine, did I? Yeah, so polymorphism on the 129 position influences susceptibility. So this is one of the amino acids and this is the other, okay? So basically, in unexposed to feast, the frequency is 0.221, 0.514, and 0.264. Okay, I'm not writing it very clearly. Okay, so suppose you wanted to look for viability selection. So what is happening? Let's just think about it. So you have a population where you have two alleles. One allele predisposes you to death. Should you indulge in mortuary feasts? Right? If you are exposed to eating dead tissue of the same species, you might get this prion disease, which will cause you to die. Right? One allele might predispose you to that, the other may not. So what do you expect will happen? No. Say it out. Okay. So suppose there are two alleles, one, this M allele and this V allele. Okay. So if you are indulging in mortuary feasts, what will happen? So this one predisposes you to the prion disease. Okay, suppose. So what will happen over time to these two alleles? The frequency of V will go to 0. Why? Because the people who have V will die, right? Okay. So how do you calculate the impact of selection? That's theoretically. How do you calculate in this society now, if you want to actually look at what is the selective pressure or differential viability of women with V, and they have to indulge in the feasts, right? Versus those who indulge in the feasts and have the other allele. How will you calculate it? Huh? So you want, this is the setup. You think that there's selection on this allele V, right? Some other studies maybe have shown you that individuals with this allele V are more likely to be susceptible to a prion disease, okay? But that will only happen in the context of eating dead bodies, right? So now if you want to actually look at differences in viability, how will you do it? How will you look for differential selection on this allele in the context of this population?
Since we have the data for the people who are unexposed to feed,
yeah,
we can find the normal allele frequencies P and Q in the population.
Yeah, you have that now.
And assume that
this is pre-selection, correct?
Yeah.
Yeah.
And with this, we can, for this set of people, 30 people, we can say how much frequency of each unit we should have had.
Correct.
If there was no selection.
Yeah.
But there is selection, then we can compare those two and we can find how much selection.
Yeah. So that's one way. Yeah. So suppose this is the frequency. So let's make it a little simpler. These numbers are a little 0.2, 0.5. This one was 0.3, but that's not. Yeah. Okay. Fine. 0.2, 0.5, and 0.3. Okay. So then, what next? You calculate? What? I can't hear, sorry. P and Q. So you basically, frequency of M. What is frequency of M? 0.2 plus 0.5 by 2, right? 0.45, right? And frequency of V will be 0.55. Okay. So now, what should be the frequency in exposed to feast? This is the observed, right? By the way, this is the observed. Right? So what should be the frequency in the, what is the expected? What's expected? 0.45 squared, which is? Huh? Point? 2025, okay. And 0.55 squared? 0.3025. And 0.55 into 0.45 into 2? Hey, you guys don't have calculators, is it? 0.49. That problem set is going to be hard if people don't have calculators.
Expected for the unexposed.
Expected if there's no selection, right? If having a V or a P or a M doesn't make any difference to your survival.
So then it's kind of expected to work.
Yeah, I mean that's expected under Hardy-Weinberg that there's no selection. Okay, so now think back to your selection table. What was that model? Remember we had W11, W12, W22, right? And we said that that's how we can calculate the next generation allele frequency, but selection has happened here. So what is this actually now? Huh? No, that's fine. Wait. Okay, before you jump there, can you calculate W11, W12, W22? Or no? How? Huh? No.
We have 3 variables and we have 3 equations. So we can
Yeah, because basically, just think about it logically, right? So this is the number which should have been surviving, right? This is the number which have actually survived. So this has been kind of modulated to become this by selection, right? W11, W22, W12, right? So you can basically use this information then to calculate what W11, W12, W22 are. How will you do it? Huh?
0.133 will be equal to W1 into 0.2 by W bar.
Yeah.
Similarly for the other two, we have 3 equations and 3 variables.
Yeah. Yeah, so you have to use W bar, W11, W22 and you can calculate it. So basically P prime, this one, oh sorry, not P prime is P V11 by V bar. That's a viability, it's not actually W11, W22. Understood, na?
P12 prime is equal to P12.
Yeah, sorry. Yeah. This is, these are this, these are this, right? This was the frequency before selection, right? This is frequency after selection, right? So you don't really need to know V bar. You have these 3 equations and you can solve and get each of the 3. So that's how you would look at viability selection. It's not actually an estimate of W bar, but it's something like that. I mean, it's not W, but it's something like that. So actually looking at selection is not very easy, right? And you also have to have had some information that you know what these alleles are. I mean, you may not have that information at all. Also, this is a very simple example where there's a single mutation which results in differences in fitness. Many times, like when we talked about the birds, big beaks and small beaks, that's a quantitative trait. where many genes might result in a combinations of alleles at many genes might result in a big beak versus a small beak. So it's going to be very hard to, so that's always been the kind of dichotomy which existed between natural history and observational based studies on selection, right? Where you would look at, say, differential predation or something like that, differential survival in the wild of phenotypes. So this is, for example, whatever, susceptibility to kuru might be a phenotype, or color on a fish, or wing coloration. That's an easy phenotype to quantify, and you might see differential survival or something like that, differences in predation, say in butterfly wing pattern or something like that. But to link that to a genetic basis is not easy. And so most of the work where we know about genetic effects of selection is done on single mutation phenotypes. A lot of the work by Hopi attempts to do that. It's not really that simple. The phenotype itself, like when she looks at the hair of the coat color of the mice, it's not that simple. There are bands of color and there are multiple genes involved. But some things are simple, like some mutations are single mutations which result in a drastic change in phenotype, and then you can potentially look at selection. Anubhav, you want to say something? No, okay. So that disjunct between selection and studying it quantitatively is often, studying it with allele frequency models is often difficult. So this paper, this recent paper, that's why it's really supposedly novel. I mean, it's not something which we didn't know, right? But it's actually showing the fate of a mutation under different environments and how the frequency increases, and matching that to population genetic data, right? Did you guys get a chance to look at the paper? No. But yeah, so basically she has this mutation, and then they have these different backgrounds, and they actually look at change in allele frequency of that mutation. I just sent it through that email, responding to that email.
So actually, I one of the earlier papers.
Huh, you did? Okay.
Days ago, just as a family,
Yeah.
mentioned this new paper that we have.
Yeah.
Most people have seen the paper.
Okay, fine. Okay, good. Huh? You are unable to open it?
You send the link,
Huh.
try to open it. I guess it's not open access.
Yeah, it's in science, so you'll have to access it from some campus. Yeah, I mean, yeah, I'm sure your campus, whichever it is, will have access to science. They ask us all to publish in science, so they better have access to it. So any other questions? I think the main thing is, these models and all are fine, but when we actually explore, how selection changes allele frequencies in the context of drift, and Kavita, I think, talked yesterday about what happens in small populations. So selection is not able to generally have a very strong impact in small populations. So tomorrow when we use the simulation package, you'll see these things, and then it'll be kind of a lot clearer. Yeah.
How do we search for systems where we can check these models? So that we'll have to find systems where there are single mutations. So we have to, so we see some phenotype in nature, and then we'll have to find out the molecular basis, and then we'll check the.
Yeah, but check the model means what? There's no. Okay, so this is a little different from I don't know, but the way I understand population genetic models, and maybe you guys can correct me, is that at least in these very basic models, unless there are violations of assumptions, there isn't a reason to, I don't know whether you need to really check them like that. You're setting them up in such a way that you're talking about a process, and it's quantitatively there's nothing to check, except that the assumptions itself may be violated, right? But you might be interested, that's why you're using the model, maybe you're interested in selection. So you're not necessarily checking the model, but you're interested in what the selection coefficient is. So you want to use the model to understand how fast allele frequency will change. But I don't think the model itself could be wrong. If it's more complex models, but this is a very, very simple model, and unless you're violating some assumptions, like I said, you don't know the genetic basis, and so on and so forth. I don't know, is that a wrong statement, Kavita? Yeah. Yeah. So if you can't explain something when you use simulations because you're exploring some new space which you don't know about, because you don't know what are the things, like if it doesn't match, then there's some assumption which you've violated. But I don't think this model per se will be wrong. There's something in the data which you have not adequately explored. But yeah, but now there's more information on, a lot more information linking genotype to phenotype. It's still very difficult. That's why, for example, Hopi's work is also actually, I don't know which paper you did, but it's based a lot on this naturalist who actually observed that there were these different mice. And because he saw it and he recorded it, she just kind of jumped on that and basically was able to develop a whole set of data and examples, case studies, which highlighted this. But without that natural history observation itself in the first place, that, oh, these look different, or there's some difference here. You have to start there versus starting from the model. I don't know whether that made sense. So you have to start with an observation. Of course, you may not be able to get to the genetic basis, but if the question is about selection, then you have to start with that and then maybe get to the model. You might have questions about mechanism, like for example on selection itself, antagonistic pleiotropy. Why are W1 and W2 different from each other? And there you could explore that like Deepa and her lab do in model systems, right? And those are questions about mechanism per se. But if you want to understand selection, then you would be interested in a specific trait. You won't necessarily, sometimes you might do it. Like for example, I just study small populations because I'm interested in what happens in small populations. But that may not even be predictable, right? It may be different every time, and it probably is. So any other questions? Yeah, sorry, you're sitting over here.
These models can maybe apply to sequence data?
No.
No.
No, in the sense, you're talking about allele frequencies. So yeah, so if there is a single point mutation which distinguishes the two phenotypes, yes. Yeah. What did you have? Can you be a little more specific? Like what did you have in mind?
Okay, so when we are comparing homologous sequences, and that ultimately results into a phylogenetic sequence.
Okay.
Like if we want that. So my question is, when we are comparing the sequences, there are bound to be differences, obviously. So
yeah.
There, can we apply such models?
Okay, so you're talking about differences between species. So yeah, so basically population genetics is talking about population, is thinking in the realm of populations. When we do phylogenetics, we are doing a slightly different thing. We are assuming that a particular sequence represents the entire species. And then we compare, we may compare multiple individuals, but we are not using population genetic inference at all. We are ignoring impacts of selection and drift and so on, because we're just talking about, we're using something like a molecular clock, where we say mutations accumulate at a constant rate, and these two species are different because of this many mutations, because they've accumulated this many mutations, and that's why they're different. But these kinds of models are not applicable in the context of phylogenetics.